Good morning, afternoon, everyone. And so for me, it's already the late night. And today, I'm Yogi and a PhD from the SIAT Chinese Academy of Science. And I'm excited to share our recent work on minimizing the genetic code using coli. Sorry, let me see the. Okay. And we all know this line that all knowing living organisms using 64 codons to encode at least 20 amino acids. Those 20 amino acids are the basic building block of life. But do we need all 64? About 10 years ago, synthetic biologists began to test this. Church's team started with a stop codon, the UAG, opening the door to the genome recoding, and then the focus moved to the science codon. Too many animal acids have multiple codons, so we can compress the code by removing some synonyms. Church plan with the blue boxes to drop two leucine and two serine and two R codons, added the UAG code to make a 57 codon E. coli. And Chin team with the green boxes, they plan to first remove two codons and added UAG to make a 61 codon string. But the more recently pushed towards 57 also as well. But I'm sorry, I have not updated here. There are also studies that use a single stop codon only. And once some codons are freed, we can reassign them to non-standard animal acids. But today I will focus on the opposite direction, which about the reverse evolution. That means do all biological systems really need all 20 conventional amino acids? Our last universal common ancestor, the Luca, already used maybe 20 amino acids about 3.5 or 3.8 billion years ago. Their order of the appearance likely traced to the chemical availability, but the site was close to the 20 we use today. So reducing this site is maybe going against the evolution, but makes it really interesting. A recent study used the phage M13 to test MET removal. They managed to delete 23 out of 24 mites, giving a strong proof of the concept. And I like to thank GCVI workshop and the Dr. Gail Kobeli for the impressive discussion last year on the fourth workshop. And they reshaped domains and RNA binding domain and the catalytic enzyme by replacing the later appearing residues with early ones. And in some designs, the functional protein used as few as 10 amino acids. So maybe function can survive radical simplification if we compensate smartly. To downsize they safely, we must find the weakest links. So we use the Blossom 62 standard substitution matrix. The higher score means the substitution seem more often and the lower risk. We then look at the BCAA pathway, the valine, the leucine, and the isoleucine. They are structurally similar and hydrophobic and often seen in the protein core. Swaps among them are common in nature and usually do not hit active chemistry. So we first tested it in the essential genes. Here you could see their positions in the genome. And in ECNR1, we succeed in 12 out of 14 tests, and in MG6053, we had 8 out of 9 partial success. We then collected the successful sites, which guided our next steps. Then we trained the sample hidden macro model style scoring to rank coding difficulty for each swipe. The lower score means either recording, so we choose the isoleucine to valine as our primary strategy. So we first try the larger scale edits, maybe about 100 KB segments. We assembled these fragments in the east and use Raxer to replace the native segments in the ecoline. Because of time, I will skip many design details, but our goal was very simple: to confirm the genotype and the phenotype, to select the 19 amino acid strains where the edit segments replace the native ones. But unfortunately, we hit errors and fitness problems. The first 100 KB strains struggle to grow. So maybe it's possible, but the fitness cost is really hard. So we then split each 100 KB region into 10 smaller fragments, maybe each about 10 to 13 KB, and tested fitness without the native counterparts. About half of the fragments worked, and here are the examples of the clean wins. But for the fragments that failed, it was hard to pinpoint the main cause. So we need to find a sharper framework to get design. Actually, in the ribosome gene, the RPSL, changing all the 3S leucine sites just killed the cell. This is the key difference between the code compression and the amino acid identity change. The latter reshapes local physics. The way out was not keep the mutating isoleucine, but to add compensatory changes in the neighboring residues to restore the structure of the function. So we then built a four-step framework. First, to learn the isoleucine replacements from the family, the genus, and the symbionts MSAS, and then we modeling the structures of the co-evolution to design the rescue mutations, and then to match the DNA expression economics to the host, and finally the host adaptation by the direct evolution. And I will walk through the gene cycle. We computed maps across the family, genus, and symbiont on the x-axis. It has leucine positions, and on the y-axis is replacements residues. Color shows the frequency, and we see across the level conserved hotspots and many as leucine to the violin or leucine events, consistent the maybe the structure rules. And then we feed the sequence into the alpha-4-3 to apply co-evolution to propose compensatory pairs and to pick the low-risk isoleucine to the valine sites. Next, we use the co-evolution to find maybe the structure of the functional to similar to the native ones, and finally we plan the and finally we will find the protein choices with the DNA, maybe prefer the JTG for the valine and the TAA at the stop codon. And we plan to direct the evolution to search a local sequence space, but the party is ongoing and we hope to share maybe next time. And thanks to Professor Dai and our collaborators in the Harris and June labs, and thanks to JCVI workshop community. And thanks everyone. Thank you for listening. And any questions? And I want to hear more suggestions. Thank you.
Yogi, this is a massive project. Are you the only one working on this? Or is it a whole team?
Yeah, maybe I'm the only one to focus on this, and someone maybe help you, but it is really a massive and hard questions to solve.
I am.
So I'm really happy to find you, and because I have seen the last year and the year before last year your workshop, and some scientists are doing the same things, and I think it's a very successful uh, things, and I'm very happy to join this meeting.
Yes, and you should be in touch with Clara Horshova's team at Charles University in the Czech Republic, given that
Yeah.
they're not trying the same amino acid, but a similar idea. And so I assume you are recoding, just recoding a piece at a time and homologously recombine them in and hope you get a living cell.
Yeah, I firstly wanted to maybe recode a large part of the genome, but it's really hard because there are many questions and we couldn't know why, and hard to realize it. So I split it, and maybe I wanted to use the AI guided and to find more protein ways to solve these problems. Yep.
And you're just focusing on essential genes because why would you do anything else? So how close are you to making a recoded ribosome, do you think?
I think the ribosome is the hardest part, and maybe we should solve this. I'm trying to maybe recall the whole TCA cycle for the E. coli, and I think maybe it's some meaningful, and we could see from the metabolism to know more about how the 19 amino acids worked for the E. coli. Yeah.
Anyone else with questions for Yogi?
Yeah.
Don't see anything, but we don't have as many people as we did a while ago. So how close do you think you will be a year from now?
I hope maybe I hope I could to solve one or two the pathways, and maybe I could finish my 100 kb of the genome, one of the least the meanest of the essential gene, and one of the most of the essential gene parts. But it's very hard because it's the most of these genes are about the ribosomes, so it's very hard. The protein don't have the I don't know how to describe it, but just one gene make me feel very difficult.
And so is your cell an isoleucine auxotroph?
Yeah.
Can it still synthesize isoleucine?
To the finalize, it don't need any isoleucine, right? The isoleucine is in the cell's wall, but it cannot see and cannot use it. It's a finalized, but just this kind of part, it also yep, it's also called synthesize isoleucine, I think. And I have think about to create tRNA that could finalize the isoleucine to the valine, but it's also very difficult to also, yeah.
But I was just thinking that if you eliminated the capacity of the cell to make isoleucine, which that might mean disabling the TCA cycle, then you could, while you're doing stuff, you could be evolving the cell to on its own replace isoleucines with whatever it felt worked best, and so that might.
Yep.
Mm-hmm.
Yep. Yes, a very good idea. And I plan to use a CRISPR-I to the genome of the TCA cycle and the BCAA pathway to define the natural synthesis and to delete the isoleucine in the medium and for the direct evolution. I think maybe it's helpful, but I have no result about this. Yes, maybe I could test it and could share more interesting results next time. And thank
Okay.
you. Yep.
And I look forward to it. And really, thank you very much for reaching out to me. I'm sorry I wasn't able to get you on the third, but next year I will know more in advance, and you and Junbao will be on my mailing list.
Thank you. Thank you very much. And hope we could get more contact and get more suggestions from you. It's very helpful.
Okay.
Okay.
Thank you very much. So with
Thank you,
that,
John.
thank you, Yogi. With that, I declare that session 1 is over, and I hope to see all of you in 8 days at 5 a.m. Pacific time. Bye, folks.